Hello students. In this session, we'll discuss one of the important infection, that is poliomyelitis. So today, we'll discuss morphology, pathogenesis, and laboratory diagnosis in the part 1. So these are the learning objectives. After completion of this session, you should able to describe the morphology and antigenic properties of poliovirus, explain pathogenesis, clinical features, and laboratory diagnosis of poliovirus, explain the type of vaccines for prevention of poliomyelitis, and differentiating features of killed and live polio vaccines. So if you see the introduction, the polioviruses, they have affinity for the nervous tissues. Affinity for nervous tissue means it has attraction for the central nervous system. It directly affect the central nervous system, which means it directly infect the brain and spinal cord. Here it causes poliomyelitis. Poliovirus strains, they are classified into 3 types. So strain 1, 2, and 3. And next, morphology. See, the size of the virus is in between 27 to 30 nanometer in the diameter. The size of virus is 27 to 30 nanometer in the diameter. It is spherical in the shape and it has the capsid. So in the picture you can see this is the capsid, and the capsid, it is made up of capsomers. So it is composed of 60 capsomers and they are arranged as icosahedral symmetry, and each capsomer has 4 viral proteins. They are called as viral protein 1, viral protein 2, viral protein 3, and viral protein 4, and they consist of single-stranded RNA as the genetic material. Next, resistance. This virus is resistant to the ether, chloroform, bile, and proteolytic enzymes of intestinal contents and detergent. So this infection or this virus is mainly transmitted by contaminated food and water, and this virus is resistant to the bile and proteolytic enzyme of the intestinal contents, which means it is not digested by the intestinal contents. So that's what it can survive, and it can cause the infection. And in the feces, it can survive for months at 4 degrees centigrade, and it can survive for years at minus 20 degrees centigrade. And next, this virus, it is easily destroyed by using of formaldehydes and oxidizing disinfectants. And this virus is it is easily inactivated by using of the heat or even by chlorination of the water. So that's what it's safe to drink the chlorinated water. And next, magnesium chloride, it to protect the virus against the heat inactivation. So that's what MgCl2 is used in the preparation of vaccine to stabilize the virus. And it doesn't survive in the lyophilization. What is meant by lyophilization? Lyophilization means storing under cold and vacuum conditions. Next, antigenic properties. So based upon the neutralization test, this virus, they are classified into 3 serotypes. Serotype 1, 2, and 3. And if you see, type 1 is most common and it is responsible for epidemics. Most of the cases are caused by the type 1. And if you see type 2 and type 3 viruses, these serotypes, type 2 and type 3 serotypes, they are completely eradicated. And one attack of this infection gives the lifelong immunity, but only against to that serotype. So suppose if serotype 1 is attacked, then it gives immunity against to only serotype 1 only. Next, pathogenesis. And see, the infection occurs only in the man. Here, there are no intermediate host. So there is no animal reservoir. So the infection occurs only in the human beings. And the children are more susceptible to this infection. Why children are more susceptible? Because children, they have the weaker immunity system, or their immunity system is in the developmental stage or constructive stage. So that's why children are more susceptible to this infection. And the mode of infection is feco-oral route. So what is meant by feco-oral route? If the food or water get contaminated with the fecal matter, if it is consumed by healthy individual, it leads to the infection. And it is the most common route. Feco-oral route is the most common route. And next to that one, inhalation, other modes of transmission by inhalation, that is the respiratory droplets, inhalation of the respiratory droplets, and rarely it is transmitted by the conjunctival tract also. It is enters through the conjunctival, and as we discussed, there is no intermediate host. So here, how the virus enters? As we discussed, the virus enters by the feco-oral route. So first of all, the virus, it multiplies in the epithelial cells of alimentary canal and in the lymphatic tissue, and from there the virus spreads to the regional lymph nodes, and from the lymph the virus enters into the bloodstream, and this is called as viremia, that is presence of virus in the bloodstream. And along with the bloodstream, the virus carries to the spinal cord and brain. Here, brain and spinal cord, they are the target organs for the virus. And after reaching to the brain, the virus multiplies in the neurons and they destroy the neurons. So as the neurons are destroyed, the lesions are mainly seen in the anterior horn cells of the spinal cord. So as the neurons are destroyed, it leads to the flaccid paralysis. So here what happens? The virus, it spreads to the regional lymph nodes and they spill over the bloodstream, which means primary viremia. After that, again they multiply in the reticuloendothelial system and then again enter into the bloodstream and they causes secondary viremia, and then it leads to the spinal cord and brain. And sometimes we can see the neural spread, which means the virus, it also spread directly through the nerves. So this is especially we can see in case of tonsillectomy. So in case of tonsillectomy, the virus spread via the glossopharyngeal nerve, which is present in the tonsillar fossa. So this is the pathogenesis. This is outline of pathogenesis. Virus, it enters by the mouth and they multiplies in the lymphatic tissues of oropharynx and in the intestines, and there they enter into the bloodstream. Along with the bloodstream, they reaches to the spinal cord and brain, and they destroy the anterior horn cells of the spinal cord. Next, clinical features. So here the incubation period is of 10 days, and the incubation period may vary from 4 days to 4 weeks also. So averagely it is 10 days, but it may take place in between, it may be from 4 days to 4 weeks. So what are the earliest manifestations? What are the earliest symptoms? So here the child suffers from fever, malaise, headache, drowsiness, constipation, and sore throat. And these symptoms last for 1 to 5 days. So here, polio infections, it is of 4 types. So the first one is inapparent infection. So in the inapparent infection, 90 to 95% of the children, they do not have any symptoms. So if 100 children are affected, almost 90 to 95% of children, they do not have any symptom. But the virus, it is present in the stool or throat, or both in the stool as well as the throat. So the virus can be isolated from stool or throat or both. And next, minor illness. So 4 to 8% of children, they develop mild influenza-like illness, like flu-like symptoms. So we can see influenza-like illness. And next, third one, non-paralytic poliomyelitis. So 1 to 2% of patients, they develop headache, neck stiffness, and back pain. And it indicates the aseptic meningitis. So some degree of aseptic meningitis. What is meant by aseptic meningitis? Viral meningitis. So here, neck stiffness, it is a characteristic of viral meningitis. Next, fourth one, paralytic poliomyelitis. So this is a flaccid paralysis. So here only 0.1 to 2% of cases, they develop the flaccid paralysis. So based upon the site of involvement, The poliomyelitis may be either spinal, bulbar, or bulbospinal. So next, cultivation. So this virus, it can be cultivated in the primary monkey kidney tissue cultures. As you know, viruses, they can be grown only in the living cells or in the cell cultures or tissue cultures. So this can be grown in the primary monkey kidney tissue cultures, and they can be grown in the vero cell lines, which means vervet monkey kidney cell line, and HeLa cell line, which means human carcinoma of larynx cell lines, human carcinoma of cervix cell lines, and HEP2 cell lines, HEP2, HEP2 cell line, that is human epithelioma of larynx cell lines. So virus can be grown in the viroid monkey kidney cell lines, HeLa cell lines, and HEP2 cell lines. And in these cell lines, we can see the CPE, which means cytopathic effect. We can see the pathological effects in the cells, which is characterized by cellular disorganization. The cells are disorganized, which means the separation of the cells and swelling of the cells and increased refractivity and lysis of the cells we can see. Next, laboratory diagnosis. So what are the specimens generally collected? So generally we collect the pharyngeal washings. So pharyngeal washings can be collected only up to 3 weeks. So for first to 3 weeks only, the virus that is present in the pharyngeal washings. And the feces sample can be collected. This is also up to 6 weeks. Up to 6 weeks we can collect the feces, brain, and spinal cord. This is autopsy specimen. And see, the virus, it is intermittent in the feces. So which means sometimes it sheds in the feces and sometimes it is not shed. So that's what 2 fecal samples are collected, and these 2 samples are collected on separate test. So they are collected on the separate days. And next, so generally we go for the sewage testing also. So sewage testing is done for detection of poliovirus, which means either wild poliovirus or vaccine virus. So this sewage testing is done under the polio eradication program, which means it is done to verify whether the transmission of infection is interrupted or otherwise the infection is ongoing. So to detect that either wild virus or vaccine virus, we do the sewage testing. We collect the sewage samples from the various places. That is done under polio eradication program. And next, transport of the samples. After collection of the samples should be kept frozen during the transport. And next, we can go for direct demonstration of the virus and see the virus. It can be directly absorbed in the stool by direct electron microscope or immune electron microscope. By using of direct electron microscopy or immunoelectron microscopy, we can directly see the virus. We can directly demonstrate the virus. And next, isolation of virus. So here the specimens, they are inoculated into tissue cultures. So as we discussed, so they can be grown only in the tissue cultures such as primary monkey kidney cells, HeLa cell lines, and HEP2 cell lines. They are commonly used. So how the growth of the virus is indicated? The growth of the virus is indicated by CPE, cytopathic effect. So this cytopathic effect is seen in 2 to 3 days. And once we can see the cytopathic effect, so we can identify the serotype, because there are 3 serotypes are there: serotype 1, 2, and 3. So these 3 serotypes are identified based upon the neutralization test by using of pooled and specific antiserum. So by a neutralization test with the pooled and specific antiserum. Next, serology. So we can detect the antibodies. So for detection of these antibodies, we need the serum sample. So serum sample, it can means we can detect the antibodies, 4-fold rise of antibodies in the paired sera by neutralization test. So what is mean by paired sera? Paired sera means 2 samples are collected within 1 to 2 weeks of interval. So means once after collecting the first sample, within 14 days, again the second serum sample is collected. And next, molecular method. So we can identify the virus by using of RT-PCR, that is real-time reverse transcriptase polymerase chain reaction. So that is used to directly to identify the like a virus in the stool specimen. Thank you. So this is part 1. In part 2, we'll discuss about prophylaxis, which means type of vaccines, and like we'll discuss about polio eradication program and pulse polio immunization. Thank you for watching.